Hello and welcome everyone to the Cells of Peace presentation I'm going to give for my final Visualizing Peace virtually integrated project, which is essentially a combination of all of the different things I've learned this semester combined into sort of a scientific communication style version of what I envision peace building to be, kind of going off the back of my last few assignments that really closely linked cell biology and the concept of peace building. So this is quite an interactive presentation today, taking you through the different stages of what a very specific cell, an astrocyte, as you see here in front of you, looks like, and how different combinations and different factors that can affect our cells can then destroy those cells and then also have the potential for healing, which is what we can classify as potentially peace building. So this is something quite subjective again, but a lot of the information is taken from scientific data, and you'll be able to actually kind of accurately visualize what is going on here. So you also have a glossary, you'll have access to kind of seeing exactly the details of what an astrocyte is after this. I'll show you that at the very, very end. But just for some kind of context for you guys. So astrocytes are essentially a very key component of our bodies, and they are a very specialized type of cell that we find in our brains. And they help to keep our surrounding cells very healthy and in tune. They're like a key kind of functional process. They're the most common cell type in the brain, and they even outnumber our neurons, which are one of the most popular cells that people know about. And they work as part of a community within our brains to make sure that there's a level of balance, or even labeled as peace, that is maintained. So in a very healthy state, astrocytes have this lovely expanded shape with complicated branches that have kind of spanning the whole brain. So together, all of these different branches on the astrocyte, as you can see, will then combine. And we call these branches, just for scientific context, peripheral astrocyte processes, and they interact very closely with our neurons, blood vessels, and other brain cells to make sure that we have a community. So you can already see the link to society and to real life if you want to kind of make that connection, as I'm trying to do here, to show that you can actually kind of compare how our cells function to peace building and to how societies work together. And just for context, this kind of cell in the middle, that is one singular astrocyte, and you can see the kind of branches spanning out. And this image, actually, I will explain in a little bit how I got to it, but it took very long to be able to code all of this. So I essentially took a microscope image and coded that and turned that into, using artificial intelligence tools, turn that into something that's quite visual, something that's not really been done before. I really struggled to find any other kind of images that looked like this, purely for illustrative purposes. And so the purpose of these cells in our bodies is going to be kind of nourishing other brain cells, so our neurons, by giving them growth hormones and energy. We want to maintain that really specific balance of homeostasis, which is essentially, you'll see in the kind of definition section, it's a combination of different systems working together with different feedback loops to make sure that we are stable. It's basically the stability of our hormones in our body. And of course, for structural support and to facilitate repair and regeneration of any damaged cells. And so the concept of this is to link the idea of cells undergoing a form of stress, just like how societies and communities would do, and visualize it as a sense of peace building. Because just like how peace building wants to kind of create collaboration, eliminate conflict, improve societal well-being, astrocytes are almost similar in that sense, in which they will support and protect this neural landscape. And so when astrocytes are becoming reactive, or if they malfunction, this piece is disrupted. So naturally there's going to be brain issues. And although it's not directly comparable, you can't really compare the two things, there's some sense of linkage and an underlying understanding that if there's one kind of cell not working and not functioning in a whole community, in a system, it's similar to how kind of one person maybe not willing to cooperate will kind of lead to disruption of a community. And so these are the kind of references. This was the image I actually had to build the healthy astrocyte cell. So I had to go and scavenge through different scientific research papers and speak to university professors from my country back home, Kosovo, and see where I could get the exact kind of tools needed. So I had to code quite a lot, use a combination of DALL-E, Stable Diffusion, which are all artificial intelligence-based image generation software. So it was not simple, but I'm glad they turned out somewhat visually appealing. And then onto the concept of sort of a life cycle is what I want to kind of showcase. So we can have our very healthy cells an initial point, but if a cell becomes partially damaged, you can immediately see the sort of difference, the comparable difference between the two types of the cell. So if we just very quickly go back to what the healthy cell looks like, you can see kind of the center is very covered. There's bulbous kind of more areas where the nucleus and the storage of the DNA is. And then you have your kind of branches spanning out. So that, of course, looks healthy. Maybe even to the untrained eye, it looks healthy. The color looks good as well. But you can immediately see the difference in where there's partial damage. So the cell, the membrane of the cell that contains all the different components to allow it to function and to grow is starting to deteriorate. And you can kind of see the different components leak out, maybe little kind of proteins coming out, or the different kind of functionalities changing because there's an external stress factor that's caused it to become slightly damaged. So there's stages of this. You'll see a healthy cell, partially damaged cell, completely damaged cell, and then a healing cell. And so just for some context, damage occurs externally to these types of cells, and the one we're focusing on is cortisol. So cortisol is a hormone that's released by our bodies when we're responding to increased stress levels. So if our brains have a lot of stress and it's just consistently being attacked by stress, there'll be high cortisol levels there. And that therefore means that the hormones, the increased level of cortisol, means that the astrocytes will actually start to lose their function because they're being consistently attacked by this specific hormone. So you'll be able to sort of notice that the outer layer of the cell is going to basically dissipate, like it's breaking the whole kind of outer layer down. There's going to be holes that are forming, and this complex network of neurons, vessels, and other cells, essentially the community that was being built inside our brains, is going to sort of retract and simplify, which of course is not going to have the best implications for us. And so there'll be some different structural changes. There'll be an alteration in the actual shape of the astrocytes. So due to the high cortisol levels, the surrounding neurons are not going to be regulated. So the other cells that send all the chemical signals and messengers in our brains will lose that regulation because that was the purpose of these beautiful astrocyte cells. So that therefore means that there's different downstream effects. For example, imbalanced hormones. So that homeostasis, that strong level of balance inside our bodies is going to be really greatly reduced. And that kind of leads to inflammation, which is where our body kind of flares up in response to some negative outside effect. And so just like how every single individual human is going to contribute into their communities, the astrocytes will essentially do the exact same, but this time for our functional brain. And there's of course going to be different factors that will contribute to this level of high cortisol. This will naturally include a whole variety of things, and you can't actually kind of pin down one specific main stressor and main kind of release of cortisol. It's a combination of different factors, but some very significant causes of high cortisol include lifestyle factors that will mean there's prolonged stress. So not just kind of one deadline and one exam and you're stressed, but consistently over a buildup of years. Things like very strict work schedules, so maybe health imbalances, relationship problems, drinking issues, life changes. Also things that really kind of expose trauma and conflict. Maybe an external conflict, being in the middle of a war zone, could then naturally trigger our body's response to high levels of stress, and that could lead to different neurological illnesses due to decreased astrocyte function and early life adversity, epigenetic changes, so things within our kind of DNA that actually change fundamentally before we're even fully formed as humans. There's an entire kind of array of factors, and it can be quite difficult to kind of attack those and reduce the cortisol levels, but it just kind of goes to show that everything really is interconnected and related, and if at our cellular level we're not kind of healthy and happy, then naturally that's going to have a negative onboarding effect as to where we're looking at peace building. And so again, this was the kind of inspiration I used for the DALI and stable diffusion image generation. All of these are taken from scientific papers and microscope images, so they are completely approved and peer-reviewed. Then kind of going on to the full cellular damage, this dramatic-looking astrocyte cell, it's completely discolored. You can see the branches have started to bulb at the end. and not kind of continue with the growth. The kind of center point of the nucleus is completely just opened out. And you can see there's a lot of damage going there. There's really visual kind of representations of it. And if we have prolonged cortisol exposure, so continuously for multiple years, maybe even a decade, that's where the astrocytes are going to undergo severe damage phase. So once we've had the highest possible level of cortisol, which is not really easy to measure, it's more based on kind of scientific data. Once we have that exact high level of cortisol, then there's going to be a phase of severe damage between 12 hours and 3 days post-exposure to this chemical. So it doesn't necessarily mean that the first time you have a lot of cortisol inside of you, that suddenly you're going to be having dead astrocyte cells. That's not the case. It's really prolonged exposure that's reached a huge threshold. And there's, of course, additional phases with this that mean destabilizing the circuits, maintaining our brain. So essentially the breakdown of our brain's cellular community, and therefore could lead to the development of neurological and psychiatric disease. And this was something I did not know about, actually. I came across this in my research here, that depression has the ability to be kind of developed from destroyed astrocyte cells. And this is really, really novel and new scientific research that I didn't even kind of have awareness of despite doing a cell biology degree. So it's really important to remember that interdisciplinary subjects will, of course, allow us to think beyond the means that we have. And kind of easing those symptoms out. So I guess at this stage, our immune system is really going to work hard to combat the destroyed astrocytes. We're not just going to have dead cells floating around our bodies. We can do things to actually help and reverse this, because cellular healing is real. There is a real kind of art and science behind it, in which our cells will then therefore come together, provided that our entire bodies are not destroyed, of course. There will always be kind of opportunities for regeneration of cells, because our bodies make so many cells. We have billions and billions of them in our bodies working for us. So if there's kind of a small clump that are getting destroyed, provided it's not cancerous, we can work to combat this. And some good techniques include stress management, improving the quality of our sleep, regular exercise, eating food that's quite rich in probiotics and vegetables, because that kind of regenerates us, meditation, of course, seeking professional support, and honestly having conversations as well, talking to people about it, kind of aligned with the concept of professional support. And that kind of aligns very well with our inner peace healing, kind of the more spiritual sense. And provided that both can kind of align together into one, we can achieve a very good state of peace, despite even being attacked by high cortisol levels. And so, as I said, the bodies will, of course, fight back. So in response to these dying astrocytes, the neurons are going to undergo a lot of structural changes to become reactive and create their defense response. So if you remember back in the beginning, astrocytes are paired very closely with this very common cell in our brains called neurons. And so provided that the astrocytes are dying, then the neurons are kind of activated to immediately clump together and produce a defensive response. And so that will kind of be the barrier to prevent it from, kind of spreading into the rest of our bodies. And then astrocytes will also downregulate their own chemical pathways, acting as feedback mechanisms to reduce our stress effects. So there's multiple things we can do there. And so this was the kind of very damaged image that was used as a combination for inspiration against quite a few others, just to showcase that there's really kind of an impact when cells are there and getting destroyed. And then the concept of cellular healing. So it's sort of a life cycle around here to show that regeneration really is possible with these cells. And a beautiful looking image where you can see the bulbs are starting to reform, the branches are coming back, there's contact once again, the center is healing as well. So very good news. There will be a repair process, and the damage is not necessarily permanent depending on how severe it is. And our cells are very, very smart, and they can achieve their own internal body peace. But there's, of course, ways that we have to kind of contribute towards that in ourselves as well, consciously. Things like lifestyle adjustments to reduce these levels and to keep them down despite being kind of deeply exposed to them. Astrocytes reversing their structural alterations by regaining their branching. So between 5 days to 1 week later, you'll kind of start to see the astrocytes are sort of regenerating. So welcome back to part 2 of this video. Apologies, it got cut off. So I'm just going to kind of take us back to the concept of cellular healing once again, remembering that we're looking now at the repair process. So damage isn't permanent. There's of course going to be ways that we can alleviate everything we've just undergone. So of course, consciously making an effort to adjust our lifestyle to reduce these cortisol levels is number one. But then astrocytes also have the ability to sort of reverse their structural alterations by regaining their branching, provided they're not undergoing insane amounts of cortisol level exposure once again. Between 5 days to 1 week later, there will be an entire time period in which they'll start to sort of regrow their paps, as they were called, the branches, and that therefore means there'll be kind of more interlinking and a restoration of these homeostasis activities because of the increased connections. And this therefore means that these cells can then neurorepair. So neurologically speaking, in the brain, there's going to be repairs by stimulating the production of neurons, since those two types of cells, the astrocytes and the neurons, work together very well. So just kind of going back to the overall diagram image to really illustrate to you what I'm thinking of, there are kind of bulbs that are kind of coming out of the branches. Those are the regeneration of the interlinking and the connections. So then looking at the concept of cellular peace. So again, very subjective, but just as peace building is going to kind of craft collaboration across the world to reduce this sort of conflict, astrocytes are the ones that are really preserving the cerebral world, if you will. So by maintaining this kind of level, our cells have the ability to heal. Then we can have a very specific level of inner peace. This is subjectively quantified by, of course, a number of functional astrocytes. In this context, we need to think about kind of what exactly we're targeting in the sense of who has been affected and what context have they been affected, how many cells do they have before and then after, and what sort of microscopes are being used. There's a lot of kind of theoretical data that needs to be drawn from this and really reinterpreted. But there's quite a few parallels that can be drawn from this. And I'd love to pose a question to kind of anyone watching out there. What sort of parallels could you draw from this version of cellular healing and peace building? And what kind of links do you envision now that you've kind of learned more about the idea of cortisol and astrocytes that you perhaps didn't before? And this was the kind of healing astrocyte image that was used as my inspiration point. So you can see there's kind of cells coming together, they're kind of growing outwards. That was quite a fascinating cell to see. All of these microscope images were really amazing. So it was lovely to be able to have the opportunity to kind of visualize those. And it actually helped me as well to see all of these different cells kind of visualize better. And so that's, I guess, the overview in the sense of having a sort of flow chart and a diagram and a visualization and sort of a life cycle as to how the cell can go from being perfectly fine to getting slightly damaged to completely destroyed and then regenerating again. That's kind of how our societies work in the same sort of system as well, with rules and regulations and making sure everything is doing well and there's plans in place. Things can be definitely avoided. So this is the kind of ending part of the presentation in which I give a little bit more context of the different components of these cells. So we spoke a lot about astrocytes. I'm well aware that a lot of you are not biology students or aware of all of the biological data that's out there, because there's a lot and it's very difficult to know. So this is just a context of you. This is what a simple astrocyte cell typically looks like. The color is more pink, but this was kind of the coding output that came out for me this time. You can see there's a nucleus, there's branches, and then we have this thing called astrocytic end feet. So these will form part of the connections that are made to the different neurons, which we class as a blood-brain barrier. So like all the different connections, the combination of cells working really hard together, that's sort of the intermediary in our bodies. So definitely the peacekeepers, I would say. And then, of course, some cellular definitions, because it's not easy to hear all this information, not understand any of it. So we'll kind of go into depth a little bit more. So an astrocyte is a type of brain cell, like we've been saying, that performs really essential activities in the body, includes sort of regulation of blood flow. allowing nutrition to reach neurons. And then a neuron, again, is another type of brain cell, but even more specialized, and they function only to send out electrical and chemical impulses for information. So when, for example, you touch something hot, the kind of cells that will actually produce a response as a result of you touching something hot are your neurons. So they'll immediately fire to your brain saying, that was hot, don't do that again, essentially. PAPs, they're the branches and the molecules that we need for chemical activities in our body. So they will kind of connect different cells together from the astrocyte. And the vessels, things like blood vessels, they're the kind of, I guess, biological tunnels, if you will. They're sort of structures in our brains that mean we can transport oxygen and nutrients to brain tissue to make sure that there's, connection with everything. Homeostasis, I mentioned this in quite a few previous projects. It's a process of sort of maintaining our stable and balanced internal environment in the body. So making sure that everything is working as it should be, kind of like how rules and regulations exist in society today. That's the kind of equivalent inside of our bodies. And then the central nervous system is quite a common part of our bodies. It's a section of our nervous system that really includes the brain and the spinal cord. And it's essentially the combination of both that will mean we function to allow chemical messengers to our bodies. Cortisol, like we said, is a hormone that's produced by our bodies. So it's a chemical that is literally generated in our glands to respond to stress. And so a hormone, there we go, is a chemical messenger, as I said, produced in the endocrine glands, so very particular areas of our body that control loads and loads of processes. Epigenetics are just changes in the expression of our genes, coding DNA, because there's different sections of our kind of genetic makeup, if you will, kind of goes into DNAs and then genes and then chromosomes. So those are all kind of wrapped up versions consisting of how we will function today, and which parts will get expressed determine who we are mentally and physiologically and kind of externally. So it's a combination of all three factors. And this change in our DNA is as a result of environmental factors only when it's referring to epigenetics. I know I briefly mentioned calcium signaling. So that's a mechanism where astrocytes will connect with our neurons to control blood flow in the brain. So these are just different kind of ways of communication. It's all a sense of communication, just like how in peace building we have different maybe webinars to kind of raise awareness for different factors, or we have kind of podcasts or social media outreach to showcase this is kind of what we do and how to keep a strong community going. That's very similar to what calcium signaling is. It's essentially a mechanism to make sure there's connection occurring. And so I love to always add a little food for thought section, because as with anything that is linking such complex subjects like biology with peace building, which are very subjective ideals, it's important to understand that you need to question sort of everything you consume in relation to media for anything, really. This is sort of a life skill to be able to do. And so, like I said here, we really need to critically analyze everything before coming to a final conclusion. So saying and showing to you guys that peace building can kind of be also interlined and interlinked with cell biology is not something that can be taken lightly. So obviously you have to critically analyze absolutely everything. And you might agree with what I'm saying, you might have a counteracting point, and that's the beauty of speaking about peace building and things like that. So analyzing advantages and disadvantages of a very specific model that's being proposed to you is really good practice to just pick apart information and not taking anything just for its face value. And that's something I really, really learned in this module. I love the concept of critical thinking, and I love to think curiously and to pick apart my world around me, but I never really came to appreciate that until I was given topics and ideas to do by myself and to explore, specifically in peace building. So it's been a very lovely time and process to actually think about what I'm writing and creating. So this is food for thought for me as well. And so I guess advantages with this learning model, the concept of cells of peace. Of course, you have increased accessibility, you're understanding biological concepts that are really complex, much simpler. And there's a lot of better emphasis when you're looking at peace building as a concept, when you're tying it to something very tangible. People can actually understand, identify, and comprehend what's going on visually, especially with the kind of AI generated images that mean people can actually see, like, wow, this is the cells that scientists would, slave away for hours looking at under microscopes, and it's here in front of me as a result of such incredible technological advancements. And so another really important point I found with this is that there's the ability to be more flexible when you're customizing what you want to craft and illustrate with this 3D model. So with the coding I had to do for Stable Diffusion, which is essentially an artificial intelligence branch where you can take text input and visual inputs and create and craft something even bigger and better. It's a very long process, and I had to teach myself to code properly because of it, and gain some support as well from scientists to confirm that it looked good. But with this, you can have the ability to add layers to your models, to change the colors, and to make the image, bigger, smaller, to really represent exactly what you want, which is great when you want to showcase something hyper specific that is not visible in the everyday world. Of course, there's disadvantages, so accuracy issues that were all generated with AI and coding tools, so it might not necessarily be the best. Since this, if you put it into perspective, the models of artificial intelligence we're dealing with now are going to be the worst versions, because every single kind of day they're getting better and there's going to be better improvements. So really important to remember and to keep in mind about that. And of course, oversimplification on complex biology of astrocytes, but also oversimplification on peace building. It's never going to be that simple as just, sit back and relax and tan in your garden and eat ice cream. So you really have to actually put into perspective and analyze it depending on where you are, and put yourself in other people's shoes as well. Potential for misinterpretation, might not be seeing the models in the way that I envisioned it, and technological limitations, as I mentioned, since AI is getting better every single day. And some afterthought questions for you to continue your research. I want everybody to use this as a platform to continue. So what insights have you obtained from astrocyte healing that could be applied to peace building? What sort of kind of visual representations have stimulated in your mind? Think of 3 key peace building strategies that could be created after you've read, analyzed, watched, and listened to this presentation. And how could we now use the current technologies of artificial intelligence, or even complex subjects like cellular biology, adapting for further steps of peace building? Definitely some food for thought and some consideration to have, since this is such an incredibly interesting interdisciplinary topic that is not really explored absolutely everywhere. And so just to wrap up, these are kind of my reference lists for further reading. I had to look at a lot of scientific research papers to digest all of the knowledge and the information down to make it simple. And also, these are where the microscope images are taken from. But if I were to kind of continue this in the future, I would love to turn this into something more physical as a product, potentially even as a storybook, and to kind of push this out further and to link it slightly more with the concept of peace building. So adding more peace building related materials and showcasing exactly how the two link. But yeah, this is my presentation for now. Thank you so much for watching.